When we study responses to global change, we're often concerned with drivers that are going to change over seasons, decades, or even centuries, which opens up the possibility that organisms evolve in response to this. Now, because of this, we might want to know what these evolutionary responses are, and one way to study that is to watch evolution in the lab.
So both of us work on marine microbes. However, evolution can occur in any organism as long as that organism has enough time for its genotypic composition to change. In the extreme, that can be from one generation to the next generation.
So when we expose organisms to a new environment right away and just see what they do in the very short term, we're often measuring a plastic response. And a plastic response is what an organism can do by changing the expression of its genes. It's just using what it has. It's today's organism in tomorrow's world. And we all understand plasticity. This is what you do if you sweat or shiver due to changes in temperature. It's what the Hulk does when he hulks out. You see big changes in traits, but no underlying change in genetic material.
So in order to study tomorrow's organisms in tomorrow's world, we need to take evolution into account. One way to conceptualize what evolution actually is to think about antibiotic resistance. So what you have here is a population made of largely of bacteria that are going to die if you feed them an antibiotic. These are the yellow bacteria. You have in that population also a couple of bacteria that are already resistant to that antibiotic. They already carry a mutation that makes them resistant. These are displayed in red. Now, if you hit that population with a strong antibiotic, most of the bacteria that don't carry the resistant-making mutations are going to die. So you're left with a population made up mainly from the red bacteria.
So what's experimental evolution? Really, it's just a method where you take populations and you evolve them in the lab under conditions that you care about, under the drivers that you're interested in, for long enough for evolution to happen. And long enough will depend on a lot of things. It will depend on how strong selection is for that population in that environment. It will depend how much standing genetic variation there is, how fast your population can generate more variation, and whether or not your population has sex. So these are all things that have to be considered when you're planning an evolution experiment. And this means that some multi-generational experiments will involve evolution and some won't. One thing that's really nice about experimental evolution is that it's meant to be general, and one way that we keep things general is by framing what's happening in terms of fitness. And fitness is quite easy to define, but notoriously difficult to measure. Fitness is the contribution of a genotype to the next generation. So it's how many of your genes you put forward into the next generation. And that means when you're planning an evolution experiment, you need to have a pretty good idea of what constitutes fitness in your particular experiment. And that's something that to some degree you can manipulate. For example, if there are no grazers in your experiment, then resistance to grazing is not part of fitness in your experiment.
One example that we wanted to find out, we looked at whether or not evolution is different in stable versus fluctuating environments, and whether or not it matters if you go into an environment being already plastic or not having a lot of plasticity. In order to answer our questions about how fluctuations in the environment have an impact on evolution, and specifically on the evolution of plasticity, we needed, and which by the way we managed to answer that question.
It was her entire PhD.
It was her entire PhD. It was like more than 2 years of non-stop experiments, but regardless, there were a couple of things that needed to be in place for that to happen. So our driver of choice needed to be one that was fairly reasonably easily manipulatable in the laboratory, and also it needed to be a driver where we knew it was going to change in the future and where we knew that it differed in different regions of the ocean today.
So that was a pretty tall order, so we ended up choosing CO2. We also needed an organism that was really widely distributed and easy to grow in the lab, because you're going to discover that we grew a lot of cultures of this organism, and that's how we fell madly in love with Austriococcus. We grew these 3 replicates of each population in 4 different environments for 400 generations, and by we, I mean Elisa. You can see there are 2 levels of CO2, ambient and high, with fluctuating and stable levels of CO2 for each case. The stable ambient was the overall control, and this is a standard lab environment for Austriococcus. The fluctuating ambient selected for plasticity only, So no high CO2, just experiencing environmental fluctuations. The stable high CO2 environment was selection for adaptation to high CO2 only, and the fluctuating high CO2 environment selected on both plasticity and the ability to grow at high CO2.
So at the end of the experiment, we used reciprocal transplants to measure how much each population had evolved. We measured some traits that were important for how Osteococcus fits into the marine ecosystem, so this could be size, photosynthesis rates, and cellular composition. But to understand what was going on with the natural selection, we measured two different fitness proxies, that being growth rate and competitive ability.
We found that populations with more plastic ancestors evolved more. And this agreed, thank goodness, with all of the theory on the topic, which gets me to the next bit, which is that experimental evolution is founded on a large body of theory, which means we often can make really strong hypotheses for our experiments and often run models and simulations beforehand, which is good because the experiments take a long time. We have a few tips for you, and the first one, probably the most important one, is to simulate your experiment and analyze your simulated data before you start. MEDDLE is a great place to start with this, but simulating experiments can be done with a wide variety of platforms.
However you end up designing your experiment, it's really important that you know your statistical power, and you can get to that statistical power from your simulated dataset. Also, really importantly, carry out pilot studies.
Tons of pilot studies.
Tons and tons of pilot studies.
Third tip is to have an idea of how large you expect your effect size to be and how noisy you expect your data to be. This is going to influence not only the amount of replication you use, but how you choose to detect your signal and the methods that you use. It's also going to affect the number of times you have to measure the same population over and over, if that's one of the ways that you're detecting evolution.
Related to power, make sure you have enough statistical replication, because otherwise you won't be able to link cause and effect.
Experimental evolution, or evolution, is sort of the zen of science in that it's one of those words that everyone throws around a lot, but not too many people are sure what it actually means. Evolution is a well-developed science with quantitative theory inside of it. So we strongly suggest collaborating.
Especially with marine biologists, because marine biology and evolutionary biology often ask questions that could actually benefit from collaborating with each other. Sadly, marine biology and evolutionary biology in the past have kind of grown up separately from each other, but we strongly suggest that you collaborate.